wanted to welcome everybody to the FRAXA Science Update. I'm Katie Clapp, and I'm one of the co-founders with my husband, who's here, of FRAXA, and our 23-year-old son, Andy, has Fragile X. If you're curious about him, we have kids pages on fraxa.org, and we have a page for Andy. So we're going to have two speakers, Becky Zorovich and Mike Tranfaglia. They're going to go back and forth with a little bit of the new and exciting updates. And then we should have some time for questions at the end.
Hey, hi, everybody. I'm Mike Tranfaglia. I'm the Medical Director of FRAXA Research Foundation. I'm a psychiatrist by training, and any of you who know me realize that we do have a son with Fragile X. So this is something that we care very deeply about, and I've been coordinating a lot of the research effort over the last 20 years nearly since we started this. This is certainly the most exciting year yet, and I was hoping to tell you about some of the recent news that's made it into the popular press in Fragile X, and we'll try to keep things relatively understandable for those of you who are not trained neuroscientists, and also try to keep things informative enough for those of you who know a little bit about the science, because one of the things that I think distinguishes the Fragile X community is we have a lot of parents who are really remarkably knowledgeable about this stuff. So the first thing I wanted to talk about is the Arbaclofen trial results that were recently published. This is just a quick scan of the Arbaclofen trial paper that was published. Now this is the phase 2 trial of the drug that Seaside Therapeutics is developing, and they've labeled this STX-209. You may recall that they previously had another drug called STX-107, which is an mGlu-R5 antagonist. That program is dormant for the time being, so this is really the lead compound for Seaside Therapeutics. It's Arbaclofen, and it's kind of the right-handed stereoisomer of the available drug baclofen. Baclofen is something that's been around for a long time. It's a treatment for spasticity. It has muscle relaxant and sedative properties. And it's a GABA-B agonist. So it is a drug that increases the function of one of the primary receptors for GABA, which is an inhibitory neurotransmitter. So they first noticed that Arbaclofen had some potentially useful properties in Fragile X by observation of patients who were just serendipitously treated with regular baclofen, who seemed to have an improvement in social functioning and language utilization. And in the autism community, apparently a number of physicians are using baclofen to treat some of the gastrointestinal problems that were associated with autism, and they just happened to serendipitously notice the other effects of the drug. But it turns out that baclofen was actually studied a long time ago by Miklos Toth at Cornell under a FRAXA grant, looking at exaggerated startle response and just general hyperexcitability of the nervous system. And he showed that baclofen could help to normalize startle response and also could treat the audiogenic seizures that we see in the Fragile X mice. And of course we see seizures in people with Fragile X too, so it's a highly relevant symptom. But there wasn't until recently any real preclinical evidence that baclofen might be disease modifying on a cellular level. But I'll get to that later, because along with this was published a companion piece that showed that Arbaclofen actually does seem to have some interesting disease-modifying properties in some of the kinds of experiments that we like to do to reassure ourselves that a drug is not just treating the symptoms, but it's actually treating some of the core features of the disease. But back to the clinical trial. In this one, on page 2, they announced the results of the clinical trial in 63 subjects. I think 60 actually made it through the trial protocol with Fragile X. And it was a crossover trial, which means that everybody got the active drug for part of the time, and then they crossed over to placebo at some point. And in the order in which they got those two would be reversed in some cases, and it was all selected randomly when you entered the trial. So some people got placebo, then they got the active drug. Some people got the active drug, then they got the placebo. A couple of the early trials in Fragile X were done this way because it's a little quicker to do. It's a little easier. You don't need as many subjects because everybody is a control and a placebo control and a drug case in one subject. So you get more statistical power from this. But there's a little bit of a complication in that when you have an unknown mechanism of action and an unknown response time with the drug, you never really know how much of a carryover effect you might get with the drug. So it kind of confounds the issues a little bit, and the subsequent larger trials that are going on now in phase 3 are using a parallel design where everybody gets placebo for the whole time or active drug for the whole time. And that simplifies the analysis quite a bit, I think. But in this case, they had a bunch of subjects in this phase 2 trial, which was designed to demonstrate the safety and tolerability of Arbaclofen in the Fragile X population primarily. And in that regard, it was a great success. The drug was very safe. No real medical problems came out. It was well tolerated by just about everybody. It didn't cause a whole lot of side effects above and beyond what you saw in the placebo group. So Arbaclofen is something that is generally easy for people with Fragile X to take. That was the primary point of this phase 2 trial. A secondary objective is to try to get some indication of efficacy. And in that regard, it was also successful, though maybe not in the way that everybody had hoped going into the trial. And you'll see that on the primary outcome measure of the ABC irritability subscale, there really was no difference between Arbaclofen and placebo. In fact, numerically, placebo was actually a little bit better. So it's not even a matter of a statistical trend without significance. It's just actually no difference at all. And then the other indicators, which I would call maybe a little bit more subjective, of the Clinical Global Impression Scale, CGI, for improvement of CGI-I and CGI-S's severity. You can see that there really was not a whole heck of a lot of difference. It didn't achieve statistical significance. There was maybe a slight hint that there was a better effect from the Arbaclofen than from placebo, but not real convincing. They also used another assessment method, asking clinicians and parents which drug, only knowing that they were on two different things, not knowing which was which, asking them which one they thought was better. And generally speaking, more people preferred the active drug. Both parents and clinicians seemed to be able to tell which one was the real stuff and which one wasn't. And they also used this thing called a visual analog scale, the VAS. And that means basically in advance you stipulate, this is the parent would say, what is the most difficult behavior you're having. And then as you go through the trial, you would just draw out on a visual scale exactly how bad that is, and sort of a sliding scale from 1 to 10. And on that, there was superiority of the active drug over the placebo. There seemed to be improvement in problem behaviors, but those problem behaviors varied from one subject to another. Excuse me. On the next page, if you look at the lethargy and social withdrawal subscale, in a subpopulation of the subjects, this is 27 of the subjects who had elevated scores on this other subscale of the ABC called lethargy and social withdrawal, you'll see that there was a trend toward improvement in this classically defined subscale of social avoidance. But more recently, David Hessel has done a refactoring of the ABC scale, which was originally designed for just general developmental disability populations. David Hessel's done a refactoring specifically for Fragile X, And he comes up with a new subscale. Rather than calling it the lethargy and social withdrawal, he just calls it the social avoidance subscale. And if you look at that social avoidance subscale, you see that there is actually a significant improvement. There's no real effect on this subscale from placebo, and there is actually a significant, and even not just statistically significant, but clinically significant improvement in this group. And you also see that if you separate out these folks that have these elevated social withdrawal scores, you then get, in this after-the-fact analysis, a better treatment effect with the active drug than you do with placebo. So the upshot of all of this is that the study didn't meet its original objectives, but its original objectives were really primarily just to demonstrate safety and tolerability, and they did that. But as far as efficacy, what they showed was that the investigators were probably looking at the wrong endpoints, and so they're actually going to start looking more at this social avoidance as an outcome measure when using baclofen. That's really what this drug does. Arbaclofen seems to help people with Fragile X to interact better socially and to utilize language better. And it's not necessarily something that helps with irritability or just general behavioral problems. There was also, as I mentioned, an interesting companion study looking at some of the preclinical paradigms that we use to test drugs in the Fragile X knockout mice. But I can just show you some of the details from that study, and that arbaclofen does some surprising things when you look at it in the lab and in the mouse model. And if you have a chance to actually dissect apart the knockout mouse brain, you can show things like, for example, on page 6, normally the knockout mice show more protein synthesis in the hippocampus than their wild-type litter mates. And if you give the wild types and the knockouts arbaclofen, what you see is that the wild types essentially have no effect. So the wild types are the black bar on page 6. The gray bar shows little or no difference between the treated and untreated conditions. But the knockouts, the red bar, have a much higher statistically significant level of protein synthesis. It's something like 32% higher in the knockout mice. And when you treat them with STX-209 or arbaclofen, that difference just goes away. It's essentially the same as the wild type. So they revert to normal with arbaclofen treatment. And that's kind of a surprising result, and that's very impressive, actually. If you look at another indicator of functioning on the cellular and synaptic level, and you look at the internalization of AMPA receptors, and these are a particular kind of glutamate receptors, different from the metabotropic glutamate receptors we're always talking about. But AMPA receptors tend to confer a certain degree of maturity on a synapse. So the more AMPA receptors you have at a synapse, the more mature it is, the stronger it is, the more effective it is. And the problem in Fragile X is that you tend to internalize too many of these AMPA receptors. They're not out there in the synapse where they're supposed to be working, but they actually are pulled out of circulation into the cell. And so you can see on page 7, versus the wild type, the knockouts have a much higher level of internalization of AMPA receptors. So that's not good. That's basically keeping synapses immature. But if you treat those knockouts with STX-209 at what would be a moderate dose, 10 micromolar, or a very high dose, 100 micromolar, what you see is that it returns back down to normal levels, wild-type levels. That's also surprising. You wouldn't necessarily assume that a GABA B agonist would have such immediate effects on AMPA receptors, but it does. These systems are all connected together. How does arbaclofen actually work on specific mouse behaviors? That's important too, because we're not just talking about cells in a petri dish. We're talking about functioning of a whole brain here. And if you actually administer arbaclofen to the mice, there are a number of behavioral outcome measures, if you will, that you can look at with the mice. One of them shown on page 8. If you look in the upper left-hand graph, you can see the results of the audiogenic seizure paradigm, which is a very robust assay in Fragile X. The wild-type mice just generally don't seize much at all, but in Fragile X mice, if you make a loud noise, they will just go into seizures. And you can test the ability of various drugs to block that. It's a really good indicator of how well something's going to work in the live animal. And what you see is that they're comparing STX-209 on the left-hand part of the upper left-hand graph to regular baclofen, the stuff that you can already get. And they're trying to show relative potency here. And what you're seeing is that the STX-209 pretty potently suppresses the seizures. The more you give, the dose is going up as you move to the right along that horizontal axis. The frequency of seizures goes down dramatically, and it only takes about 1.5 milligrams per kilogram to get rid of the seizures for the most part in the mice. That would be considered the average effective dose to decrease the seizures by 50%. And that seems to be more effective than even 2 times as much, 3 milligrams per kilogram of the regular baclofen. Racemic means it's a mix of R-baclofen and R and S baclofen, the right and the left-handed versions of it. So that tells you 2 things. The R-baclofen is effective in one of the primary preclinical measures of efficacy in a Fragile X mouse, but also regular baclofen is effective. It's just that it takes at least twice as much to do the same thing. So that's something important to consider, that if this never makes it to market, we still do have regular baclofen that we can consider using as a potential treatment, and it does seem to have some useful properties. They did a number of other behavioral tests and basic lab tests, but the basic point is that our baclofen is showing disease-modifying properties in a lot of the basic kinds of lab tests that we use to validate all sorts of different compounds. And keep that idea in mind, because the assumption here is that it's very difficult to show in a clinical trial that a particular drug fixes Fragile X rather than just alleviating specific symptoms, because inevitably when you're doing a relatively short-term clinical trial in human beings, you're just looking at behavioral symptoms. You're looking at outcome measures that tap into easily observable behaviors. You're not able to dissect apart brains and look for molecular changes, but we can do that in the animal models. And so if we test these compounds in the animal models and they show that they have these really potent disease-modifying effects, and then we take them into clinical trials and we see really good behavioral effects, we can feel a lot more confident that we're getting actual disease-modifying effects from these drugs rather than just alleviating symptoms of Fragile X. And with that, I'll turn things over to Becky, and I think she's going to talk a little bit about some of the new research that we're doing and some of the kind of final common pathways in Fragile X and autism that are being identified. Becky?